Thank you, Koei. Thank you for the introduction. And thank you all for being here. Also want to say thank you to the dkNET pilot program for the opportunity to speak today and for funding a lot of this work. So I'm excited today to tell you about the work that we've been doing to understand leptin action on sort of a single cell level. And as I'm sure we're all aware, obesity rates and its related comorbidities such as diabetes have been increasing throughout the decades. This is in part because we lack very effective treatments right now to counteract obesity. Now, we know that obesity risk alleles are associated with genes that are expressed in the nervous system. So we believe that targeting the brain is going to be our most effective way of developing treatments for obesity. Now, this involvement of the brain in human obesity was not necessarily a surprise because we've known for a long time from animal models of monogenic obesity, sort of most famously here in the leptin deficient mouse line, the OBOB mouse line, that monogenic forms of obesity are also mediated through the brain. We use leptin as a system to understand cell types and biological processes that are important for maintaining normal energy balance, in hopes that these cell types and processes can be used for future therapies. Now, leptin is famously secreted by adipocytes in proportion to triglyceride stores, and it acts on cells that express the leptin receptor in the hypothalamus to restrain food intake and promote energy expenditure. Now, if we were to zoom in on the hypothalamus, we can see that this is actually a very complicated structure with dozens of subnuclei. The most famous of which is the Arcuate Nucleus down here at the base of the hypothalamus. And the Arcuate Nucleus contains many distinct cell types. Two of the most well-studied are the AGRP cells and the POMC cells. Both of these cells respond to leptin, but in opposing fashions. So leptin inhibits AGRP cells, and AGRP cells promote food intake. Leptin stimulates POMC cells, and POMC cells suppress food intake. And this leptin to arcuate AGRP POMC mechanism has been the predominant source of study in the field for many years. However, we also know that the leptin receptor is broadly expressed throughout the hypothalamus. So this is, for example, just one coronal plane through the hypothalamus. You can see the arcuate down here, then also the ventromedial hypothalamus, dorsomedial, and lateral hypothalamus. And there are many leptin receptor populations that are likely unidentified. As a field, there's been a concerted effort to identify molecularly defined populations of leptin receptor cells. So, for example, many groups have identified unique populations in the VMH or the DMH, LHA, and so on. However, we know that the cell types that we have identified so far do not encompass the entirety of the leptin receptor populations in the hypothalamus. And we also know that deletion of leptin action from these cells does not recapitulate the extreme obesity that you see with total loss of leptin action. So our goal is to identify the complete set of leptin receptor cells in the hypothalamus and be able to uncover novel populations that are important for energy balance. So we know that leptin mediates its effects predominantly through a JAK-STAT signaling pathway, where leptin binding to the leptin receptor results in phosphorylation of the transcription factor STAT3 and transcription of STAT3 target genes. We had previously shown by deleting STAT3 in leptin receptor cells that the transcriptional effects of leptin are almost completely mediated by STAT3. So for example, STAT3 versus OBOB, bulk RNA-seq is very similar, except for a huge chunk of STAT1 target genes because of compensatory upregulation of STAT1. So we know that STAT3 and in a compensatory role, STAT1 mediate the vast majority of leptin transcriptional effects. However, to date, this has only been done in bulk tissue, and we don't know if this differs on a single cell level or population level. Ultimately, in states of obesity where leptin levels are chronically high, we know that exogenous leptin becomes less effective at mediating its hunger suppressive and energy expenditure promotion effects. So for example, excessive levels of leptin drive baseline high levels of phospho-STAT3, as well as feedback inhibitors, ER stress, and interactions with neighboring glia that can be detrimental to the normal leptin response. This is what's known as leptin resistance. And this is famously shown here, where in baseline conditions, there's very minimal phospho-STAT3, but then is dramatically upregulated with leptin treatment. And then is almost completely lost in animals that are maintained on high-fat diet, where leptin levels at baseline are chronically high. So we also would like to know, what are the sort of molecular mediators that are resulting in this loss of STAT3 sensitivity, and whether or not this differs by cell types, and maybe some cell types are more susceptible to leptin resistance than others. This motivates sort of our major questions here, which are, I've sort of outlined somewhat quickly, are, can we identify the complete set of leptin receptor populations in the hypothalamus? Do they have distinct leptin signaling pathways? How are these impacted by obesity? And then ultimately, if we're going to develop any of these findings into therapies, we want to know if the populations and the transcriptional responses are conserved in primates, including humans. So to address all these questions, we've turned to single-cell transcriptomics. This field has been rapidly advancing with becoming much cheaper and able to profile many more cells. So the beauty of single-cell transcriptomics is that we can take an unbiased approach of all the cells in a given chunk of tissue. We can use clustering approaches to identify cell populations with no prior knowledge. And then based on these cell types that we identify, we can find genes and transcriptional motifs that are distinct to given cell types. Now, one of the issues with a lot of the sort of droplet-based forms of single-cell sequencing is that they tend to be very low depth, and the leptin receptor itself is a very lowly expressed gene. So, for example, I'm just showing you here an early published single-cell RNA-seq dataset, one of the sort of pioneer datasets looking into the hypothalamus. And you can see they recover all the major cell types, of which neurons are about half. But then when you look in more detail, there's virtually no leptin receptor detection, because leptin receptor just happens to be more susceptible to dropout using these droplet-based single-cell approaches, so that about 1% of the total cells in this data set contain the leptin receptor. This is obviously going to be not useful for us to be very confident in identifying cell populations that express the leptin receptor. So to circumvent this, we have been using an enrichment approach. So we borrowed this idea from Jeremy Nathan's group, where they created a mouse line in which there is a Cre-dependent expression of a nuclear envelope GFP. So SUN1 is a nuclear envelope protein which targets the superfolded GFP to the nuclear envelope. The idea being that we can take this mouse line, we can cross it with our leptin receptor Cre, and this will label leptin receptor nuclei with GFP. And you can see that's exactly what happens here. So now I'm just showing you a few different sections throughout the hypothalamus. You can see that areas of the brain that we know express the leptin receptor have robust expression of this nuclear GFP. And actually, if you were to zoom in, you can see these tiny little donut-shaped holes for the nuclear envelopes, labeling the nuclear envelopes. So now that we have a mouse line that labels leptin receptor cells with GFP, we can use FACS sorting to enrich for them. So in this series of experiments, we've taken this mouse line, leptin receptor SUN1 GFP, dissected out the entire hypothalamus, isolated the nuclei, used FACS to enrich for GFP, and submitted them for droplet-based single-cell sequencing. And then we run these the results through a sort of standard analysis pipeline, which involves quality control and then embedding in a lower dimension, in this case UMAP. So what you can see is that as opposed to about 1% of the cells expressing the leptin receptor, I'm now showing you for all the cells we've collected, about half of them express the leptin receptors. So we're very strongly enriching for leptin receptor expressing cells. And instead of about half of the cells being neurons, we now have almost 95% of the cells being neurons, with a few glial cell types identified as well. We believe these are most likely FACS artifacts, cells that were sorted that are not truly leptin receptor positive, because as you can see here, these cell types do not really express the leptin receptor either. But now that we have this data set, we can use unbiased clustering to identify cell types. And what we see is that when we take just the neurons and cluster those, we get many dozens of distinct leptin receptor cell populations. You'll probably notice some of these are ones I've already discussed. For example, we identified the Arcuate HERP cells, the VMH NR5A1 cells, the Arcuate POMC cells, and so on. However, there are also a wide variety of novel populations that have yet to be studied. And then we then sorted these by leptin receptor expression. And what we found is that while AGRP and POMC are among the most highly populations with the highest levels of leptin receptor expression, we identified a novel population that has marker genes enriched in the dorsomedial hypothalamus and expresses the GLP-1 receptor that has the highest levels of leptin receptor. It also is a very abundant population, more abundant than AGRP and POMC. So we became interested in this population. But we next wanted to know, now that we have this data set in hand and this mouse line in hand, we want to know on a population level how different leptin signaling is. So in addition to just doing unbiased sequencing, we also treated some animals with leptin, so either vehicle or leptin for 6 hours. And you can see we've taken the same approach of isolating nuclei and performing the same pre-processing approach. You can see here that the vehicle and the leptin-treated cells are well mixed, meaning we don't have major batch effects that would prohibit us from identifying clusters or treatment effects. You can also see that across all populations, we have fairly balanced treatment groups, indicating that we're able to find robust effects of treatment. So we next wanted to know which populations have the most differentially expressed genes. And to control for differences in cluster size, we measured differential expression from 100 random downsamplings of the data to keep each single population the same size. And what you see here is that, again, this high leptin receptor population that expresses the GLP-1 receptor has the most differentially expressed genes in the vast majority of its downsamples. However, again, POMC, AGRP are up there, but there's also a variety of other leptin receptor populations that are robustly regulated by acute leptin. We also analyze this data in a different way because this requires you to first do clustering, and clustering can be somewhat arbitrary. We then used an approach which uses nearest neighbors. So the idea is that you have a control and a treatment. If you have a cell that has a lot of neighbors from the treatment group, it is most likely more similar to treatment than it is to untreated. And then we can use this, and given the known proportions of treatments in the data set, we can say what the likelihood is of that cell being affected by treatment. And you can see here now we've plotted it across the same UMAP plot. And again, you see these DMH GLP-1 receptor cells are highly biased towards association with leptin-treated cells, indicating that these cells are highly regulated by treatment. Again, we see POMC cells are, but less so AGRP and a variety of other populations here. This is essentially another way of showing what we've shown previously using the downsampling approach. Plotted another way, you can see here that the leptin cells are very likely to have leptin neighbors in this GLP-1 receptor population, indicating that they are highly regulated by treatment. That is not necessarily the case for all other populations. So we became very interested in this DMH GLP-1 receptor population, has high levels of leptin receptor, and it's highly regulated by leptin treatment. So we wanted to know precisely where they're expressed in the hypothalamus. So using two distinct approaches, we generated mouse lines where we could co-localize leptin receptor-driven GFP and GLP-1 receptor expression, or GLP-1 receptor-driven GFP and leptin receptor action. And you can see in both cases, we see that overlap of these two genes is localized to the dorsomedial hypothalamus, and predominantly in this sort of stretch here in the anterior as well as the ventral dorsomedial hypothalamus. And as you can see here, there's essentially no overlap in the arcuate nucleus. So we next wanted to know what role these cells play in leptin action. So we deleted the leptin receptor from these GLP-1 receptor cells using the GLP-1 receptor pre. And we found that these animals gain weight, about 20% of their body weight. And that is driven exclusively by increases in food intake. I'm not showing you here. We've also measured energy expenditure. We found that that's totally normal in this mouse line. So this indicates that we've identified a novel leptin receptor population that is resident in the DMH, and it is critical for leptin's effects on food intake and ultimately on body weight. So now using this data, though, we wanted to know what are the factors that are driving the association with of leptin effects. Why is leptin treatment different across populations? One of the first things we found was that if you just take the average leptin receptor expression and the number of differentially expressed genes, you see there's a fairly linear relationship. So leptin receptor itself seems to be associated with effective treatment. What's fascinating is that aside from just sort of an aggregated level, this is true also on a single cell level. So if we take leptin receptor expression on a single cell level and the probability that a given cell will be associated with leptin treated cells, you can see that I've colored it by cluster here. Within given clusters, cells are more likely to be leptin responsive if they have higher levels of leptin receptor. This probably sounds somewhat obvious, but that was not necessarily what we expected going in, because different populations have different downstream mediators expressed at different levels. They have different availability of circulating leptin, presumably, and potentially different levels of baseline phosphostatin reactivation. So the fact that leptin receptor is a major driver is very interesting, and allows us to sort of zero in on populations that could be important. And lastly, I'll show you here on an individual population level, you can see the very nice correspondence between leptin receptor expression in very distinct subsets of each population and the probability that these cells are highly regulated by leptin. But now looking broadly across all the populations, we wanted to know how similar or different leptin signaling is in these distinct populations. So I'm showing you here the correlation of fold changes across the various leptin receptor populations after treatment by leptin. And you can see that there's a block up here in which that are very highly correlated, indicating that they have very similar responses to leptin treatment. And this block up here corresponds to sort of the classic leptin receptor populations, AGRP, POMC, the VMH, the GLP-1 receptor cells. And these are the ones that have the highest levels of leptin receptor expression. And if we were to just look, for example, at sort of these classic leptin target genes, you can see that they're all coordinately regulated in these sort of classic leptin receptor populations, indicating that leptin drives a very stereotypical transcription cascade in each distinct cell population. But what is leptin doing was our next question. So what we decided to do is we took the regulated genes and we ran them through gene ontology analysis to look for enriched terms. I'm showing you here a plot of all the regulated terms with their associated p-value across all populations. What you'll probably notice is that there's this little chunk in here that is consistently regulated in every single cluster that we looked at. And when we zoomed in here, what you can see is these are all associated with synapse assembly, postsynaptic membrane, and associations across the membrane, indicating that leptin, acute leptin treatment is driving acute changes in synaptic function and potentially in synaptic contacts. When we looked at the genes that are in this synaptic GO term, what you can see is that virtually all of them are robustly downregulated, indicating that leptin treatment is dramatically changing the makeup of synapses in leptin receptor target genes. This is also somewhat interesting because leptin has been shown for a long time to be critical in development for promoting and maintaining appropriate synaptic contacts. But we're now seeing this in the adults, indicating that leptin could be important for synaptic machinery in the adult. This has been shown, for example, in hippocampal neurons, where leptin is important for promoting AMPA receptor activity associated with long-term potentiation and depression. But this could also be a major mechanism, a transcriptional mechanism in the hypothalamus as well. And this gives us this sort of handle on the genes that we can go after to understand what the precise molecular changes are. But now we wanted to know, what are the mediators that are actually driving this response? We know that leptin receptor itself is sort of predictive of the response, but what are the intermediate mediators? So we turn to an approach where we can identify, using our single cell data, transcription factor networks that are active in individual cells. This is an approach called CNIC that was first developed a few years ago. So we took our data set and established epigenomic data to identify transcription factor motifs that are active in the leptin receptor populations. What I'm showing you here are the hundreds of distinct transcription factor regulons on this axis and individual cells on this axis. And what you'll probably appreciate is, across the vast majority of transcription factors, they are very consistent across cell types as well as across treatments. You can see there are a few unique transcription factors that are biased towards specific states or specific populations. And when we looked at the vast majority of these, we found these tend to be transcription factors important for cell identity. So for example, NKX 2.1 was a major active transcription factor, and NKX 2.1 is known to be important for hypothalamic identity. You can see here that it is very strong in all cells, but it is not. However, we then took all regulons and we found their association with treatment. And what we found after reducing them in dimensions to better visualize them, we found that a very similar batch of regulons here that are the sort of STAT and STAT target genes. So for example, STAT3, STAT1, STAT2 are all very highly associated with leptin treatment, as we would expect. But then also another chunk of transcription factors, such as JUNB, ATF3, and EGR2, which are known immediate early genes, indicating these are leptin is actually activating populations as we would expect. Now, when we look in more detail at the STAT3 regulon, we can see that while some populations have baseline high levels of STAT3 activity, this essentially across the board goes up very dramatically with leptin treatment, as we might expect. And as then we can now pull the predicted STAT3 target genes from this analysis. And what we see is that across the vast majority of the sort of classic leptin receptor populations, there's a coordinated change in all STAT3 targets. The exception is these sort of very small populations that have very low levels of leptin receptor, indicating that leptin treatment, again, across our populations is very stereotypical and driven predominantly by STAT3. So now that we know sort of the baseline leptin transcriptome, and we've identified a wide variety of leptin receptor populations, we wanted to know what's happening to leptin signaling during metabolic states such as obesity. So to address this, we turned to, instead of using our SUN1 approach, we turned to using wild type mice that were maintained on either chow or high-fat diet. The reasoning is because now that we have our leptin receptor populations, we can identify them in the broader hypothalamus in an unbiased way. And also we expect a variety of high-fat diet effects to be on non-leptin receptor populations, and we don't want to miss those. So this in this data set, we have now 20 samples from across the hypothalamus, including some micro dissections of the arcuate VMH and DMH, encompassing about 187,000 cells. You can see there's good depth, well mixed by sample, and well mixed by treatment. We were able to identify all the expected cell types in the hypothalamus. And one thing to note is that we did not see a major impact on cell type composition with high-fat diet treatment. It's possible that there are maybe changes in subpopulations of some glial cell types, but on the whole, we don't see major shifts in cell type proportions. We then can take this and identify neuronal subpopulations. And we find that there's very extensive neuronal diversity in this data set. And each of these populations encompasses chow high-fat diet treatment, and then varies, of course, by neurotransmitter use and are biased towards distinct dissection regions. So to identify what these populations are, we then turn to using some published data sets as a reference atlas for labeling our data set. So I'm showing you here of some example data sets that we use, and you can see that correlate populations tend to have distinct expression across our data set. And then we can use this to then label our data set with established populations, as well as some sort of novel populations in the middle here. And you can again see we have VMH out here, including our VMH leptin receptor cells, AGRP cells out here, cells from the paraventricular hypothalamus, and so on. We then wanted to know what are the effects of high-fat diet on this data set. So we just ran differential expression analysis across our entire data set. And again, we see that these DMH GLP-1 receptor cells are extremely highly regulated by high-fat diet, as are the AGRP cells. When we look specifically at the transcriptional response here, we found that a wide variety of the genes that are targets in a high-fat diet are leptin target genes. The same was broadly true for AGRP cells. So that led us to ask, are these responses in high-fat diet leptin responses or something else? Now, just a reminder that leptin receptor itself is very poorly detected by these approaches. So I'm showing you here leptin receptor expression in our data set, and it's a tiny fraction of the total cells. But we can use our SUN1 data set to find cells that are correlates in the broader hypothalamus of our SUN1 populations. And you can see here, this allows us to pull out a wide variety of cell types, for example, out here, that do not have any robust leptin receptor expression itself. And that allows us to then label our broader hypothalamic data set with their established leptin receptor population. And comfortingly, we see that our leptin receptor SUN1 populations have essentially one-to-one correlates in the broader hypothalamus, with the exception of sort of these unidentifiable sort of GABAergic glutamatergic populations in the middle here that are likely aggregates of a wide variety of hypothalamic populations. So we wanted to know, does high-fat diet treatment look like leptin treatment? So we then took our leptin receptor SUN1 populations and correlated the effects of leptin treatment with the effects of high-fat diet treatment in the broader hypothalamus. And you can see that for the most part, there is no correlation across the vast majority of populations. But there is sort of a modest correlation in this small chunk of populations out here. And when we looked to see what those are, you can see that, again, these are sort of our classic leptin receptor populations, DMH, GLP-1 receptor, POMC, VMH, and so on. However, we wanted, in a more sort of principled way, we wanted to know, does an individual cell in the high-fat diet treatment look like a vehicle cell, or does it look like a leptin cell? So to do that, we integrated our leptin receptor SUN1 data set and our broader hypothalamus data set. And then we could use nearest neighbor analysis to say, are given cells, are they more likely to be a vehicle cell or a leptin cell? And what we found when we looked at our sort of classic leptin receptor populations is that if you look up here for the GLP-1 receptor cells, vehicle cells are almost always predicted to be, or chow cells are almost always predicted to be vehicle cells. High-fat diet cells are almost always predicted to be leptin-treated cells. And this is what you would expect if leptin is still driving its transcriptional responses in high-fat diet. But now if you look here down at AGRP cells, you can see that vehicle, chow are predicted to be vehicle, but high-fat diet cells are also predicted to be vehicle cells, indicating that some signal of leptin action is lost in high-fat diet in AGRP cells. And that's even clearer when you just sort of look at the correlation of differential expression in each cell population. So for example, in the GLP-1 receptor cells, there's a somewhat nice correlation in treatment in high-fat diet and treatment with leptin. But basically, in AGRP cells, the effects are completely uncorrelated, indicating that there's basically two distinct transcriptional responses that are happening in these two treatments. And when we looked more specifically, we can take those STAT3 target genes that we'd learned from the scenic analysis and look at their fold changes by our various treatments. You can see the GLP-1 receptor cells with leptin treatment and high-fat diet have very similar transcriptional responses. AGRP cells have robust transcriptional response of STAT3 targets with leptin treatment, but effectively in high-fat diet, the STAT3 target regulation is almost completely lost, indicating that these AGRP cells are the likely leptin resistant cells that you can identify in the arcuate nucleus by loss of phospho-STAT3. But then looking beyond just STAT3, we can look at all transcription factor networks that were identified as regulated by leptin or regulated by high-fat diet. And what you might notice here is that here is the level of regulation with leptin across the x-axis and the level of regulation with high-fat diet across the y-axis. And across all these populations, there's a much more pronounced regulation with acute leptin treatment than there is with high-fat diet treatment. That's somewhat not the case for the GLP-1 receptor cells, where STAT3 is still robustly regulated in both. However, you can see for AGRP cells, essentially all leptin-regulated transcription factors are unchanged in high-fat diet. And if we just order that by the difference between their activity in high-fat diet versus leptin, you can see that STAT3 and STAT1 are dramatically less active in high-fat diet than they are in leptin treatment. And the same is true of other very strong leptin target transcription factors that we learned from our previous SUN1 analysis. So now that brings us to sort of the next approach, which is we wanted to know, so now we know that we've identified a wide variety of leptin receptor populations in the hypothalamus. We know what sort of the baseline leptin transcriptome looks like. And we found that while leptin action is similar in a variety of mouse populations, the susceptibility to leptin resistance does vary by population. But the nice thing about single cell approach is that we can use tissue from a variety of different organisms to identify the same approach, identify the same transcriptional effects. So in collaboration with Paul Kivett, we have been using tissue from macaques that were maintained on either a chow or high-fat diet. And Paul's group has obtained tissue samples from the brain bank and microdissected either the arcuate, VMH, or DMH. And you can see here we have a few animals from covering a wide variety of ages and sexes and body weights. So our analysis would not be biased to some sort of small set of animals. Now, we have run essentially 15 samples here. You can see we get great depth from these brain bank samples. And then using standard approaches, we're able to unite and harmonize all of these data sets into a single data set. We can then learn the cell types using our mouse data set. And you can see we recover all the major cell types we expect from the mouse hypothalamus. They all have robust transcriptomes and marker genes that, classic marker genes that we know from the mouse are also highly enriched in the macaque, indicating that the macaque hypothalamus looks very similar to the mouse hypothalamus on sort of a broad cell type level. When we then just look at expression correlation between the mouse cell types and the macaque cell types, we find that the sort of ortholog cell types are very similar. So as you would expect, neurons quite similar to neurons, microglia quite similar to microglia, and so on. So this is very comforting and means that what we learn in the mouse is likely applicable to the macaque. And especially important that neurons are sort of the most highly similar between the two species, and sort of all the classic neuron-enriched marker genes are also highly enriched in the macaque as they are in the mouse. One notable difference, though, is that cell type proportions tend to differ between species. So the mouse hypothalamus tends to have much more neurons and fewer oligodendrocytes, whereas the opposite is the case in the macaque, where there are fewer neurons and many more oligodendrocytes, indicating that the macaque hypothalamus is likely much more myelinated than the mouse. These differences in cell type proportions can't be explained by differences in, say, age or sex or treatment, because across the macaque, we did not see any obvious association with either age or sex or treatment with these distinct cell type proportions. We think the differences are likely due to either biological or potentially technical variation between the species. We then, as we did for the mouse, we can subcluster the neurons. What you see is that our arcuate samples cluster out here, DMH samples down here, and VMH out here, indicating that the region of origin is a major driver of cell type specification, as you would imagine. These populations are well covered by treatment, and then we can cluster these as we had for the mouse to identify macaque hypothalamic populations, and we recovered many dozen distinct populations. To identify whether these populations are orthologs of the mouse, we can then use the same approach we have in our previous mouse data set. We can use a Seurat CCA to integrate the samples and project mouse labels onto our macaque. And what you see is that the mouse and the macaque populations are essentially one-to-one correlates across our data set, with some exceptions, again, these sort of like indistinguishable GABA glutamatergic populations in the middle. But for the most part, the populations that we expect to see, including AGRP, POMC, NR5A1, MCH, and hypocretin, have very distinct populations in the macaque that are very similar to the populations of the mouse. Comfortingly, also, leptin receptor is broadly expressed in our data set and is highly expressed in populations that we would expect, including AGRP, POMC, and so on. Now, we want to use our broader data set to determine how conserved these populations are between the two species. So as you can see, leptin receptor itself is actually very robustly detected in this macaque data set. And however, it is confined to a small set of populations. So again, we can learn what these populations are, in this case, using the ScanV approach and our SUN1 data set. And we can label them using our mouse SUN1 population labels. What you can see here is that the mouse SUN1 orthologs of VMH are present here, POMC, AGRP, DMH, GLP-1 receptor here, indicating that individual cells and broader populations in the macaque have very clear orthologs to our mouse, which means that we can use our mouse data set to infer responses in the macaque. And if we're to sort of look at very specific marker genes, you can see, for example, AGRP marker genes here are robustly enriched in the mouse AGRP population, as well as the macaque AGRP population, GLP-1 receptor markers, GLP-1 receptor markers. So even down to an individual gene marker level, these populations are very similar. But then we wanted to see, because we have animals that were either on chow or high-fat diet, we want to see how their transcriptional responses differ across populations. And you'll see again that some populations are highly responsive and others much less so. And again, among the top populations are these DMH GLP-1 receptor cells. And now when we looked at the specific genes that are regulated in high-fat diet, we could see a variety of genes that we've seen previously. However, there was not a very clear sort of linear correlation between effective high-fat diet in the macaque GLP-1 receptor cells or the mouse GLP-1 receptor cells. And this is sort of an example of what we saw broader in the hypothalamus. So I'm just showing you here the correlation of gene expression changes in high-fat diet by the mouse and the macaque, and don't have to really take any messages home here other than there's no obvious pattern here. Correlates across data sets are modestly correlated, but nothing very clear. So we decided, because the difference between species is likely not a simple sort of linear correlate, we wanted to turn to a non-linear approach. This is a method developed a few years ago called SCGen. And the idea is that you can take cells that were treated and cells that were not treated, combine both and encode them in sort of a common latent space. you can learn the effective treatment and then take that learned effective treatment to apply it to these naive cells to predict their effective treatment for individual cells. So in our case, we can take our mouse vehicle and leptin cells, combine them with the chow. We can estimate the leptin effects, and then we can infer leptin responses in the chow cells. I'm just showing you the mouse here as sort of an example. But our goal then is to be able to use this to learn leptin responses in our macaque data set, because we can't treat macaques acutely with leptin. And what you can see here is that I'm just showing you now STAT3 activity or STAT3 expression. You can see STAT3 is predicted to be highly upregulated in all these classic leptin receptor populations, as we would expect. And it is predicted to be less upregulated in the AGRP and the POMC cells, but still robustly upregulated, say, in our GLP-1 receptor cells in high-fat diet, indicating that this approach is learning that the AGRP cells and the POMC cells are somewhat leptin resistant, whereas the GLP-1 receptor cells are less so. We can then take this approach to then actually generate a quantitative measure of leptin resistance by saying how similar are high-fat diet cells to these pseudo-leptin cells, these generated leptin cells versus these chow cells. And what we found is that across the board, most high-fat diet cells are more similar to chow than they are to pseudo-leptin treatment. It's not exactly surprising, because you would expect chronic leptin is different from acute leptin. But then we can use this to generate, to quantify how leptin resistant these cells are. And we see that AGRP and POMC, again, are the most, have the most suppressed leptin response in high-fat diet compared to a wide variety of other leptin receptor populations. So then we can take the same approach. Now that we know this approach is robust, we can take the same approach where we use our learned macaque ortholog cells. We can then embed them with our mouse SUN1 data set, estimate the effects of leptin, and infer their leptin responsiveness. And when we do that, you see again that AGRP cells are predicted to highly upregulate STAT3, POMC cells are as well, but we do not see an upregulation of STAT3 in high-fat diet relative to chow in these two populations. The opposite is true for these GLP-1 receptor cells, where they're predicted to upregulate STAT3 with leptin treatment, and we see an upregulation in high-fat diet, indicating just as in the mouse, AGRP and POMC are likely leptin resistant in the macaque in obesity, whereas the GLP-1 receptor cells likely maintain some level of leptin responsiveness. That's all I'm going to show today. So I just want to close by some sort of some major points from our study so far, which is that we now have identified many dozens of leptin receptor populations. And we know that this is a very complex population structure. There are many novel populations, but that most of them are shared in the macaque, indicating what we learn in the mouse is likely applicable to primate biology. We've also learned that leptin action, at least acute leptin action, is very similar across cell types, and that the vast majority of leptin signaling beyond just STAT3 is blunted in chronic obesity. We also showed that some macaque cell types show signs of leptin resistance very similar to the mouse in chronic obesity, indicating that we can use this data set to infer populations that would be good to go after for therapeutic targets. In the future, we'd like to measure some of these effects more directly. So for example, we'd like to treat our leptin receptor SUN1 cells with acute leptin and see if maybe AGRP or POMC cells still retain the capacity for leptin signaling. And we'd also like to know what happens to leptin signaling and leptin action across the cell types with known leptin resistance, known leptin sensitizers, for example, celastrol. Can we restore distinct aspects of leptin action, and what effects do those have on physiology? I just want to close by thanking everyone, especially everyone in Martin Meyer's lab. Abby and Ali helped a lot with the mouse models and the sequencing. And especially want to thank everyone in the dkNET new investigator program. and other funding sources. And I'm happy to take any questions. Thank you.
Yeah, thanks, Alan, for the great presentation on leptin mechanism. Now is our Q&A section. So you can unmute yourself or use chat function to ask question. Yeah. And then during the presentation, I think there's one question from the audience. It said, do you see any changes in glia in your high-fat diet treatment data?
Yeah, that's a good question. So big picture, we didn't see any changes in cell type proportions of glia. It is possible that sort of the expansion of reactive microglia, reactive astrocytes is small. They're a small proportion of the total. So we're missing that when we sort of aggregate them. That's something that we're sort of actively studying right now, but it was essentially outside the scope of our focus on leptin for this talk, but it would be very interesting to go that route.
Let me check if there's other questions. The second question is, do you think the populations that are less leptin resistant are still contributing to leptin action in obesity?
I see. So like so for example, the GLP-1 receptor cells that still respond to leptin in obesity, are they still contributing to suppressing weight gain? Yeah, I think that's possible. It seems plausible. So for example, a variety of groups have shown now that if you acutely inhibit leptin action in obesity, you then see a further increase in food intake and weight gain, indicating that some cell populations are still responding to the excess leptin to suppress food intake and weight gain. We don't know what those populations are. But that's sort of why we want to do the studies with acute leptin treatment in obesity, to be able to identify those populations that are still responsive and might be able to suppress weight gain in obesity.